Bom dia. Obrigado, Carlos. Thank you very much, Carlos, for everything you've done and all the people here welcoming myself and Melanie to Brazil, to Recife. It's been a wonderful time that we've been here. Today I want to talk some about our work in honor of Peter Morgaine, my dear friend and colleague, Carlos, and Pilar's dear friend and colleague, that we've done over the past number of years, looking at how protein malnutrition exposure during the prenatal period can lead to reprogramming of the brain. So I've dedicated this to Peter Morgaine, who is a dear friend and colleague. We talked about him yesterday quite a bit. Here we are at our university and having a celebration. He always loved champagne, and we celebrated with champagne quite often. This is the group that I've been working with Peter over the past 25 years or so. Janina Gower has been a dear friend and colleague for all this period of time. She has been a leader in the area of prenatal protein malnutrition, not only in the work that we do, not only in the work that we do in our animal model of protein malnutrition, but she has also been working with patients in Barbados for 40 years, looking at the effects of the protein malnutrition on those patients. Jill McGahey is a dear friend and colleague at the University of New Hampshire in the United States. She does a lot of the work in the behavior and the effects of prenatal protein malnutrition and behavior. Doug Rosine has been a dear friend and colleague at Boston University for many years. He is a neuroanatomist par excellence, widely recognized as one of the top neuroanatomists in the world today. Jerry Rushmore is another colleague. I'll be presenting some of the data that he's worked on this morning. And then Anna Amaral, who is is Brazilian. She did a lot of this work when she was at Boston University as a graduate student. She's now doing her postdoctoral work at the Mass General Hospital at Harvard University. And then I always have to thank all of my students and colleagues that have worked with us for many years. Donna Bass was a student as well as a technician in the laboratory. Here are some of the students as well as others that we've had over the years working on these projects with us. We couldn't do it without them. So, why are we looking at this in animals? We are looking at this in animals to better understand what is happening in our human populations that have been exposed to prenatal protein malnutrition. Some of the key findings that we have from our human studies are that in the Dutch famine study, the Dutch people right after World War II had very little to eat, and they were exposed, a large group of the population were exposed to malnutrition at that time. And scientists have been actually studying them for many generations now. And one of the key findings that they found was that if you are exposed to prenatal protein malnutrition, you have a much higher risk of being depressed, of having major depressive disorder. Also, in the 1960s, there was a period of time in China where they had famine and were not able to adequately receive amounts of protein. And in the studies that we've done with patients that were exposed to this during the famine in China, we had an increase with schizophrenia. Now, those of you that have studied psychiatry know that there's a lot of similarities about how we understand schizophrenia, major depressive disorder. They both involve higher order functions in the brain. Now, Janina Galler has, as I said, for the past 40 years, studied the effects of prenatal protein malnutrition in a cohort of patients in Barbados. And she's followed these patients for many years. She's actually doing work now in the F3, the third generation of offspring from the patients that were originally exposed to prenatal malnutrition. So she's done this for 45 years. There's now over 200 patients that she's following that are offspring of the original cohort of patients. We know that there's an increased risk of depression in this group of patients. But more importantly, from our perspective, is that much more higher risk of attentional problems. This is not attentional deficit disorder as we usually define it, because there's differences in the population study, but they do have attentional problems. And you'll see that this has been a major focus of our work in our animal model of malnutrition. So myself and colleagues have been working on an animal model of prenatal protein malnutrition. We basically are looking at the offspring of female rats that have been exposed to prenatal protein malnutrition prior to and during pregnancy. We use behavioral assessment of tests of attention. Jill McGahee is an expert in the area of models, behavioral models of depression, of attentional problems, and she's been working on that. And we focused a lot on the activity of the hippocampus and the prefrontal cortex in these different models. We also use neuroanatomy techniques to assess the birth of cells. One thought would be that the birth dates of cells may be delayed as a result of prenatal protein malnutrition. We look at metabolic activity throughout the brain. How does the brain function? Usually this is now in adult animals that have been exposed prenatally to malnutrition. But how is that influenced with the metabolic activity in the brain? Using techniques called 2-deoxyglucose as well as CFOS, which is an immediate early gene. My laboratory does in vivo microdialysis, which I spoke about a lot on Wednesday. This is basically where we're able to put probes into the brain and assess the release of neurotransmitters in the brain during behavior while the animal is awake. And we focus those activities on the hippocampus and the prefrontal cortex. And then more recently, we've started to look at epigenetics. So now epigenetics is a very important piece of what we're looking at in terms of how the environment can influence genes. So epigenetics is basically where the DNA is modified by a number of different routes in order to change the way the genes are expressed. So this is something we always think of the gene as something we are inherited, that we are born with. But then our life experience changes the way that our genes are expressed, so that protein malnutrition probably does change quite a bit of the genes that we have and causes a difference in the way that those genes are being expressed. And I'll show you some data about that later on. So, looking particularly at neurotransmitter systems with prenatal protein malnutrition, we've shown a lot in many, many different studies, not only in our laboratories, but in other laboratories throughout the world, that one of the big findings that we see is a large increase in serotonin, 5-hydroxytryptamine, 5-HT, in the brain of animals that have been exposed to the prenatal protein malnutrition. We've seen increased levels of serotonin from birth up until postnatal day 250 in all areas of the brain, the lungs, the stomach, and the heart. And this is some work by Stern et al. In 1975. And in another paper at the same year, they showed that postnatal day 110, they saw increases in serotonin and telencephalon in the brainstem, as well as decreases in norepinephrine in the telencephalon in the forebrain. These are the first papers in 1975 by the group with Dr. Morgaine, looking at prenatal malnutrition. So we're now looking at 45 years of research along these lines. But when we look at the birth dates of the cells, okay, when are the cells born in the brain, we did not see any changes. Okay, the Raphe nucleus, where the serotonin or the 5-HT cells live, as well as the locus coeruleus, where the norepinephrine neurons are. There's no difference in the birth date. So that's not what we know, that's not part of the problem. However, when we look at brains of adult animals that were exposed to prenatal protein malnutrition, we see that serotonin fibers are decreased in the hippocampus, transporters are decreased, and 5-HT1A receptors are also decreased. In selective areas, CA1 and CA3, important areas of the hippocampus. Also, there was a decrease in serotonin release when it was stimulated in slices of the hippocampus from prenatal protein malnourished rats. Also, they saw increased release of serotonin and dopamine from hippocampus slices of prenatal protein malnourished rats. So, Dr. Chen did these experiments in the 1990s. And then also, as reviewed by Sebastião Almeida, who worked with Sheena Galler in Boston, one of the many people from Brazil who went to Boston and worked with Dr. Morgaine and Dr. Galler, saw different, very altered responses to a number of different types and classes of drugs in animals exposed to prenatal protein malnutrition. Now, my technique, which I've used in my lab for the last 30 years, has been to look at in vivo microdialysis. And this is a technique whereby we put a probe surgically implanted into the brain in selected brain areas. We perfuse the probe with an artificial CSF that's very similar to extracellular fluid in the brain. And as that fluid is going through this microdialysis probe, if there's concentrations of neurotransmitters around the probe, it'll be taken up into the cerebral spinal fluid. We then can use a very highly sensitive, high-performance liquid chromatography to assess the levels of the neurotransmitter in the dialysis. So in our early studies, we were looking at the prenatal protein malnutrition effects on serotonin in the hippocampus. So at this time, we were focusing our work on the hippocampal formation, doing neuroanatomy to assess the changes in the structure of the hippocampus. We were doing behavioral tests to look at learning and memory and the effects on the hippocampus. And then microdialysis to assess how the serotonin is being released in the hippocampus. So this is the effects that we see if we put the probe into the hippocampus. As you can see here, after a period of time for that probe to equilibrate, we see a very large increase in the amount of serotonin that's in the extracellular fluid in the hippocampus. And if we then look, and this is in young rats, 30-day-old rats that are just pre-adolescent. The dark circles here are in malnourished animals, and this is in well-nourished if we give them a dose of a benzodiazepine, 4-diazepoxide, okay, this is an anxiolytic drug, then we can see a decrease in the levels of serotonin from this level up here down to normal levels, as opposed to the effects of the well-nourished animal. We then, around this period of time, in the early mid-1990s, we changed our focus. We had done many years of research on the effects in the hippocampus, felt that we had reached sort of an end of the era of understanding what's going on in the hippocampus. We had this information that we thought that there were attention problems. So now we were beginning to understand the prefrontal cortex. And we've known for many years about prefrontal cortex in humans and some of the importance of the prefrontal cortex in humans in terms of planning, motivation, being able to plan our lives and be able to function. And at this period of time in the 1990s, we were also debating in neuroscience, did a rat have a prefrontal cortex? And so we've done a lot of reading, and Peter Morgan and I have published papers on the prefrontal cortex of the rat, and we feel like that part of the brain in a rat also functions very much in terms of motivation, planning, as an executive function, cognition. And so we changed our focus from the hippocampus into the prefrontal cortex because of these changes that we saw in attention in people that have been exposed to prenatal protein malnutrition. So now we looked at extracellular dopamine and serotonin in the medial prefrontal cortex, continuing to look at the dorsal hippocampus. And in this situation, when we looked at the right prefrontal cortex of the rat, we saw that there was a decrease in basal release of dopamine that occurred. We also wanted to look at the effects of stress. So we have found, looking at behavior, that in many cases, animals exposed to prenatal protein malnutrition may look fairly normal in behavior, but if you then stress them somehow, then they start to have problems. So many of us know stress is a real factor in psychology. And so if we have some deficit in our brain function, then if we become stressed, we may have a problem with our ability to function properly. So we were looking at this change in dopamine. We didn't see any changes in serotonin, and in this case, for a change, we didn't see any changes in serotonin in the dorsal hippocampus. Now, looking at the dopamine, I graph this as two different ways. The percentage of control, so 100% is the basal release we see after 3 hours. And you can see that in well-nourished animals, we see a fairly significant increase in the levels of dopamine in the prefrontal cortex when we stress these animals. After the stress is finished, it returns to normal, and actually in prenatally malnourished animals, it falls well below normal. And then this is just looking at the absolute levels because we saw this large change. You can see the large increase in the well-nourished animals, whereas the malnourished animals doesn't respond to stress. Now, we then went on to do some additional work looking at attentional set shifting, the behavior that we see as one of the attentional tasks that we work on, and looking at metabolic activity in the prefrontal cortex. Okay, how does that change? And then this is looking at the behavior in the trials to criterion. This is the number of reversals that we see in this particular test. And as you can see, it takes more trials to criterion in the malnourished animals than the well-nourished animals, indicating it takes longer for them to complete this task. They have more time than it takes for them to do this. This is work by Joe McGahee in 2014. We then looked at the metabolic activity. This is measured by 2-deoxyglucose. 2-deoxyglucose is a chemical, it's radioactive. You can inject it intraperitoneally, into the rat before they perform some kind of task. So we injected these rats with the 2-deoxyglucose, and then it's taken up by cells that are active. And so cells that are using more oxygen, or more glucose will take up this 2-deoxyglucose. And so they'll be more active and they'll have more radioactivity. And you can see that up here. So the brighter the color, the more red the color, the more the activity. Okay? So during this attentional task, you can see that the cortex of the rat is highly lit up, olfactory cortex as well. And this is a well-nourished animal. This is a malnourished animal. And you can see that there are some changes, and that's reflected down here in different areas of the cortex. This is the anterior cingulate cortex, infralimbic cortex, prelimbic cortex, and the olfactory cortex. That you're seeing these changes. So here's the olfactory cortex here, ACC, PRL, and infralimbic here. Whereas in some other areas, like the SC and the piriform cortex, that you don't see changes in the activity. So they have less activity in the prenatally malnourished animals during this particular task, indicating that there's some deficit there. Now we've gone on to do some additional work, and this is the first paper that we've published in many years that looked both at the clinical studies and epigenetics in comparison to animal studies. So we're looking at both animals and humans in this particular study. Now this is basically looking at the cohort of patients that are in Barbados. So here's the first generation early malnutrition that's occurring here. The intervention that occurs 1 to 11 years. So basically these children were identified when they came into the hospital, when their mothers were giving birth to them, as being malnourished. And they were immediately put on a good diet. The mother was put on a good diet to make sure that they had adequate nutrition right after their birth. Okay? And now they're being, we've followed them along, done a lot of research behaviorally, testing them constantly over time. Here's the beginning of the birth of the G2, the second generation, also following them across time. And then they've been collecting DNA collection. I can also say that right now, Janina is working with colleagues in Cuba, also have access to patients with prenatal protein malnutrition. And they're now starting to analyze EEG recordings that were done early on in both cohorts, the second generation and the first generation, to see if the EEGs, how they might be different. So this is now looking at the animal work in particular. So this is some additional work where we showed looking at another attention task. Here you can see that the malnourished animals that are in red here show deficits in this attention task as a decrease in the number of hits that they have in that particular task. If we look at malnutrition versus control in the lateral prelimbic area, which is this area right here, we can see there's a high correlation between malnourished animals and the amount of 2DG is very low in the malnourished animals as opposed to the well-nourished animals. And here you can see that reflected in these graphs up here. So again, we're seeing a decrease in activity in important areas of the prefrontal cortex. in these animals. Now, we also looked at methylation. So a colleague, Shakram Varian, now at the Icahn School of Medicine at Mount Sinai in New York City, looked at histone methylation. So I talked about epigenetics. And so methylation of histones is one way that this epigenetic effect occurs. This is a pretty complex diagram, but basically it's showing the difference in human subjects between malnourished and well-nourished. All right, so the well-nourished controls. And you can see all of these marks in red are where there's a hypomethylation of histones. Those are genes that are hypomethylated. And then green is all of the hypermethylation. Now, hypermethylation can mean many different things. It doesn't mean that, but it usually means some kind of difference in the way the gene is being expressed. And when we went in and looked more carefully, we did find some very key neuro genes, genes related to brain function that had been changed as a result of prenatal protein malnutrition. And we showed in some cases that we actually had changes in protein expression that was a result of prenatal protein malnutrition. So we're finding more and more about how prenatal protein malnutrition can actually change our genome. And that's what we're looking at, and Janina's looking at in her second generation and now some third generations, because when you have epigenetic changes, if you have epigenetic changes in germ cells, okay, either in eggs or sperm, those can be translated and given to the offspring, okay, to this next generation. But if that generation now is okay nutritionally, you may lose the effect of the epigenetics in the third generation. Okay? So it may be something that is carried over in 2 generations, but may not be a problem in the 3rd generation. Now, here's some additional work using microdialysis. Here we were looking at prenatal protein malnutrition again in the prefrontal cortex. And one of the pretty things about microdialysis is that you can analyze whatever is in that extracellular fluid. If you have an analytical technique that allows you to look at just serotonin, you can do that. My technique, I can look at serotonin, dopamine, and norepinephrine in the same sample. Okay, so I get an idea of what's going on in these animals across those 3. Other people, one of my Pilar's students was working with me, and he was interested in vasopressin. And we actually did some work with microdialysis looking at vasopressin to see what changes might be happening. We were looking at suprachiasmatic nucleus and seeing circadian changes there. Anyway, going back to this. So we also did this, we analyzed these samples for serotonin, dopamine, and norepinephrine. We also looked at behavior in these animals, because all of these animals have been behaviorally characterized and put through these behavioral tests for us to be able to see correlations between them. So in this particular study, if we look at the left prefrontal cortex, we saw an increase in serotonin, no changes in the right. Now, if we look at dopamine, we don't see any changes in the left prefrontal cortex with dopamine, but we see significant changes in the right. So now we're starting to see that depending upon whether we're looking at the right or the left hemisphere, we have differences that may be important to how the brain is functioning. And no one really has started to look at the differences between the hemispheres. So this is called laterality, and why this occurs, we know it occurs in the human brain. It does occur in rat brain now, we know that. And the function of that is going to be very important for us understanding how the brain works. And then if we look at norepinephrine in the prefrontal cortex, again, the right prefrontal cortex has lower levels of norepinephrine in malnourished animals as well. So one of the things that I've done is started to look at this laterality and come up with something I call a laterality index, okay, which is basically taking the left versus the right, and so you see where the balance is. Where is the most of the neurotransmitter? And you can see here that in the well-nourished animal, norepinephrine and dopamine are very much shifted towards the left. So if it's going downwards, it's more left more than right. And if it goes upward, it's right versus left, okay? And you can see that it shifts in the malnourished animal. Again, this might be reflective of reprogramming of the brain. There's a change in the circuitry within the brain that's bringing about this change. So in summary, in terms of this particular paper, we saw increases in serotonin in the left hemisphere, but not the right. And alternatively, we saw decreased dopamine in the right versus the left in the malnourished animal. So there's this shift in laterality from the left side to the right side, from well-nourished animals to the malnourished animal. Okay? And again, we still have a lot of work to do to understand what this means in terms of behavior. We also looked at, in a group of 6 animals in each group, they had been tested in a behavioral test of attention before we had done the microanalysis experiments. And we did a correlational analysis between the level of neurotransmitter in the brain. You can see there's serotonin left, serotonin right, dopamine left, dopamine right, norepinephrine right, and the number of reversals it took them to complete the task. So how well did they do in the task compared to their, the levels of the neurotransmitter in the brain? And you can see here that in well-nourished animals, in the extra-dimensional shift, which is one portion of the task, we had an increase in serotonin, and that was correlated positively, okay? So if the animal took more trials to finish the task, they had higher levels of serotonin, right? And also, importantly, in the malnourished animal, if we look now at the norepinephrine in the right and the total reversals, in the well-nourished animal, there is a significant correlation with low levels of norepinephrine. So if they had low levels of norepinephrine, they did worse on the task. All right? So we're looking at this. So in summary, with the neurotransmitter changes that we've been able to determine, in all of these different studies with malnutrition, we've seen this kind of effect here. With the hippocampus, mostly we see an increase in serotonin. Medial prefrontal cortex decreases in the right for norepinephrine and dopamine, an increase in the left for serotonin. I'm not showing you data today from the orbital prefrontal cortex, which we've also done, where we saw increased levels of norepinephrine in both left and right in the malnourished animals, but no changes in dopamine or serotonin. Now, we've also looked at the effects of norepinephrine on attention in the prefrontal cortex and prenatal protein malnutrition. So we just published this in Frontiers in Neuroscience, and what Jill had done here in these studies is she looked at animals exposed to prenatal protein malnutrition, but she also did a study lesioning norepinephrine neurons in the prefrontal cortex. So she put in a toxin, sapotoxin for norepinephrine, in the prefrontal cortex and reduce the amount of norepinephrine fibers. And you can see her staining over here. This is a well-nourished animal versus a malnourished animal, and the darkness of the staining gives you an indication of this. This is the norepinephrine transporter, and then this is her lesion study. So this is the lesions, and this is her sham lesions. So this is now looking at areas of the brain. This is in the malnourished animal. You can see decrease in norepinephrine fibers in the prelimbic area. And then her lesion studies, which she targeted the prelimbic area, a decrease in norepinephrine fibers. But in the anterior cingulate cortex, another area of the brain, prefrontal cortex, that wasn't targeted, we don't see a change in malnourished animals. We don't see a change with the neurotoxin. And when she then does behavioral assessment in these animals, she shows that lesioned animals as well as malnourished animals have problems in completing the task. They differ a little bit depending upon what part of the task they're having a problem with. This is the lesioned animals have a problem with the predictable distractor, but not a problem with the unpredictable distractor, whereas the malnourished animals have a problem with the unpredictable distractor. So it's somewhat differences in terms of the total view of the behavior, but they're both having problems with the behavior. So we know norepinephrine is very important for attention, and we're seeing that whether we have a lesion of the norepinephrine fibers or we have prenatal protein malnutrition. Now we're looking at, we've just started to do a study, and we're working on getting this published now, looking at network analysis. So now this is more computational neuroscience, where we're looking at the relationship between 35 different areas of the brain, okay, and the activity based on this 2-deoxyglucose in malnourished versus well-nourished animals. So basically what we've done is we've taken the brains from 8 malnourished animals and the brains from 8 well-nourished animals, sliced them completely through, analyzed 30 different regions of interest within that, mostly prefrontal areas, and looked at the relationship between activity in these different areas and how those areas are connected to each other. So this is the connectivity, okay? So here's our 35 areas of interest, okay? And if you see a dark circle, there's a connection between those two areas, okay? The darkened boxes are connections between the areas. And I think, rather than a lot of detail going into it, the main thing is just look at the pattern between the two, okay? Anybody can look at that and not even know what we're doing here and say there's a difference. You know, these are our malnourished animals here. Very different than when you look at the control animals. All right? Now, of interest is this is the prefrontal area right here. And as you can see in a control animal, they're very highly connected. Okay? A lot of connectivity there between the prefrontal areas. Prenatal malnutrition, not so much. Okay, they're not highly connected to each other. They're more down here. And down here is more areas like the hippocampus, okay? The reunion nucleus, the habenula, much more connectivity in another area of the brain. So this sort of was the first indication that we had, looking globally at the areas of the brain, that there was really a reprogramming, there was a change in the entire circuitry of the brain. So we've been looking at these different areas kind of in isolation. You know, the prefrontal cortex, the hippocampus, you know, okay, we see changes here, we see changes there, what does that mean? We don't know. This is starting to tell us that there's something bigger going on. These are global network measures, so these are measures of calculations of the Characteristic path length, how closely to each other the areas that are connected are. And you can see that there's a significant difference between prenatal protein malnutrition and control. The prenatal protein malnutrition animals tend to have to go further to get the connectivity. Global efficiency is decreased in the prenatally malnourished animal. And the mean clustering coefficient, another measure, global measure, is much lower in prenatally malnourished animals. So lower efficiency within the brain. And there's some other things to look at. Local clustering coefficients, between centrality, local efficiency. And without getting into a lot of detail here, I think that you can just look at the patterns that you can see here in these graphs to see that there's quite a bit of difference. The malnourished animals are in the lighter shade here, and this is this area up in here is the prefrontal cortex. So you can see the control animals have much higher connectivity, local clustering coefficients than the malnourished animal. This again is right versus right in the. This is the infralimbic, right infralimbic cortex. Has much higher between the centrality in the control animals, not much at all in the malnourished animals. Same thing over here. Okay. So in summary, we know that prenatal protein malnutrition alters brain chemistry. Serotonin is noted throughout the brain and periphery compared to controls. Dopamine and norepinephrine are decreased in one hemisphere of the prefrontal cortex. We don't know how they're altered in other regions of the brain. Decreases in norepinephrine in the left PFC are correlated with behavior in the sustained attention task. Lesions of norepinephrine neurons in the prefrontal cortex produce similar behavioral effects to PPM. And assessment of brain networks shows significant changes. So the networks of PPM animals do not seem to rely so much on prefrontal cortex hippocampal connections. If you look at all of those connectivity, we see a lot of connections in the well-nourished animals between hippocampus and prefrontal cortex, not so much in prenatal protein malnutrition. There's a disconnect there. So it's hypofunction of the prefrontal cortex and malnutrition. So in conclusion then, reprogramming is a different idea. Okay? It's a different idea than just saying prenatal protein malnutrition causes these changes in the brain. In my mind, reprogramming means that there's been a sort of a change that is planned, or it's a change in response to the malnutrition that changes the behavior of the animal. So when we look at behavioral effects in these animals that are prenatally protein malnourished, we see a behavioral rigidity. So what does that mean? That means that they're hyper-focused. They're actually do better on the tasks. When we try to distract them, they do well. Okay, they do better than controls because they're hyper-focused on that task. All right? So they're hyper-focused in order to be able to focus on getting better food, getting more food, getting better nutrition. Okay? And we still need to do more work to understand this reprogramming. So, obrigado, thank you, and I'll be glad to take any questions.
I actually do have a question. It's more like curiosity, and it's about your rat results. Have you ever looked at sex differences?
Well, That's a really good question, because that's something that comes up every time we start to work on our things. So you saw when I presented my data on Wednesday that we looked at microdialysis in female rats. We see very big differences in these neurotransmitters in female rats as compared to male rats. And unfortunately, every time when we go through these studies, there's so many components and things that we get to the point of saying, we need to do females, we need to do females, we need to do females. And I think the only way we're ever going to do that is if we just do females. We're not going to be able to do a study that says, here's males, here's females, here's malnourished, here's well-nourished. Because it's just going to be too many animals. We can never figure out how to do it. You know, the NIH, when they give us these grants, they give us many dollars, but it goes very quickly, it seems, you know, and time to do all of these studies, to do the numbers that you need to. So, yes, we really want to do female animals, but we haven't done it yet.
Yeah, because the differences between sides, right and left, are impressive. So I was just wondering if that's only in males or if the females can be the same, or maybe
it seems to me when I think back to that study we did with the microdialysis and prefrontal cortex in female animals, the differences between left and right weren't as big as in the male animals. If I remember correctly, I'm thinking of the graph that I showed. And dopamine in female rats was much higher in females than in males. Same with serotonin, but it seems to me that there wasn't a big difference that we saw one side to the other. We saw a bigger difference one side to the other in adolescents versus adults. Yeah.
Outro.
Obrigado.
Devido ao tempo, né? Vamos. Gostaria de, em nome do NeuroNutri 2019, ofertar uma pequena lembrança ao Dr. David Mokler. Peter Mokler